Assalamu alaikum. This is I am going to discuss here the mechanism of enzyme linked receptors. In this mechanism, receptors are actually itself act as enzyme, or they get attached with some kind of enzyme. Here example is the leptin receptor. This leptin receptor is present as a dimer. Dimer means it consists of two units, first unit and the second unit, and this is attached to the enzymes known as Janus kinase 2, JAK2. Okay. Uh, these receptors have protein hormone binding site present outside the cell membrane and enzyme binding site present inside the cell membrane. Okay. As soon as the leptin gets attached with this receptor, there is a cascade of chemical reaction occur inside the cytoplasm. And the Janus kinase 2 enzyme belong to the family of the cytokines, and its phosphorylation occur. Here you can see the phosphate group gets attached with the Janus kinase 2. Then it activates. Then this Janus kinase activates another protein that is known as STAT3. Its phosphorylation occur, and then this STAT3 protein enter into the nucleus, where it gets attached with the target genes, and its target genes are present on the DNA of the nucleus. Here then transcription occurs. As a result of the transcription, messenger RNA is formed, which is released outside the membrane of the nucleus through the walls, and then messenger RNA undergoes translation. Translation is the process of protein formation, and actually this protein formed during the translation is the physiological effect of the cell. Janus kinase 2 activates enzymes, and enzymes are responsible in the formation of the protein, and this is the final result of the cell. How these receptors functions. Leptin is the hormone which is released by the fat cells, and it is mainly involved in the regulation of the appetite. Next topic is the intracellular receptors in the target cell. Previously, I discussed hormones, which are protein, peptide, or catecholamine in nature. They require special type of the receptors present in the cell membrane for entry into the cell membrane. However, the hormones which are lipophilic in nature, they do not require any special kind of receptors present in the cell membrane, as you can see in this diagram. Lipophilic hormones have either receptors present in the cytoplasm or inside the nucleus. First of all, lipophilic hormone can easily diffuse from anywhere in the cell membrane, because cell membrane is made up of lipid bilayer, and the hormone has the same nature as the lipid bilayer, so it is very easy to cross the cell membrane. Now this hormone gets attached with the cytoplasmic receptor, and then this hormone cytoplasmic receptor enter into the nucleus, or this lipophilic hormone directly enters into the nucleus and gets attached with the nuclear receptors. Then this hormone receptor complex gets attached with the DNA on a very specific portion known as hormone response element. This leads to the formation of the messenger RNA, and now this messenger RNA leaves the nucleus, and this is the process of the translation. Ribosomes are involved in this process, because ribosomes are the protein synthetic machineries, and this protein is the physiological response of the cell. Next mechanism is the adenylyl cyclase cyclic AMP second messenger system. Here, why AMP is known as second messenger? Because first messenger is the hormone which activate or which initiate or which stimulate the reaction. Then all next steps are carried out with the help of cyclic AMP. This is similar with the previously discussed mechanism. This is the receptor present inside the cell membrane, present in the cell membrane, and some part of it is present outside the cell membrane. When the hormone from the extracellular fluid gets attached with this receptor, there will be conformational changes. G protein gets attached with this receptor, and GDP is converted into GTP, and then alpha subunit from the G protein gets attached with the enzyme known as adenylyl cyclase, and this enzyme is responsible for the breakdown of the ATP into the cyclic AMP. This activates another enzyme which is already present in inactive form in the cytoplasm, and that enzyme is known as inactive cyclic AMP dependent protein kinase. Now this is activated into active cyclic AMP dependent protein kinase, and it is responsible for the phosphorylation of the proteins plus ATP. So one phosphoryl group gets attached with the protein, and this is the they gets attached with the protein, and this is the phosphorylated protein plus ADP is formed here, and this phosphorylated protein is the cell response. This cyclic AMP mechanism is very important in physiological processes. You can see at various steps in various reactions this cyclic AMP plays always its primary role. Okay, this is the next mechanism in this mechanism. Cyclic AMP do not act as a second messenger system. Instead of cyclic AMP, here phospholipase C enzyme act as a second messenger. When the peptide hormone gets attached with the receptor site, then the same process occur. G proteins are present here, and alpha subunit of the G protein contain GDP. Then these G proteins now activates phospholipase C. This is the enzyme, and this enzyme is responsible for the breakdown of the phospholipids present inside the cell membrane. Here you can see all process occur inside the cell membrane. While in the previous mechanism, reaction was occurring in the cytoplasm. Phospholipase C is responsible for the breakdown of the phospholipids. These are the phospholipids. Okay, this is the head of the phospholipid, and this is the tail of the phospholipid. The phospholipid, this PIP2 is known as phosphatidylinositol biphosphate. It is broken down into two second messenger products, IP3 and DAG. IP3 is the inositol triphosphate, while DAG is the diacylglycerol. IP3 activates IP3 mobilizes the calcium ions from the endoplasmic reticulum. As all of you know, endoplasmic reticulum is the storage site of the calcium. When the calcium is released into the cytoplasm, this causes the contraction of various cells, as we previously discussed in the muscle in the chapter of the skeletal muscle. Calcium ion gets bind with the troponin and causes the contraction of the muscle, while DAG causes the activation of the protein kinase C enzyme into the protein kinase C active form, and this active form is responsible for the phosphorylation of the protein which is previously formed on the on the rough endoplasmic reticulum. So the response of the cell is the phosphorylated protein plus contraction of muscles, either may be skeletal muscle or the smooth muscle. In case of the smooth muscle, specific protein known as calmodulin is present.